Bây giờ này sinh viên chưa vô. Hello, Dr. Batiban.
Yeah. Just close it. Yeah.
Yes,
Can you hear
I
me?
would like to introduce some information about you. Is okay?
Yeah. Yeah.
Yes. Dr. Anbu Rajan Batiban, he's a professor, a research professor of the Department of Environmental Engineering, Seoul National University of Science and Technology, Seoul, South Korea. He's a graduate biotechnology with a PhD diploma in Bharatiya University, Tamil Nadu, India. And from the 2011 to 2013, he graduated biotechnology in the Bharatiya University, Tamil Nadu, India. From the 2014 to 2018, he graduated PhD about civil engineering with the Daegu University, South Korea. He has more than 15 year experience with the research about the environment. Now he would like to present his research about his research with the fermentative biohydrogen production from macro algal biomass using various reactor configuration. We would like to invite to you present your presentation, please.
Yeah. Thank you. Thank you so much, Professor Chow. So
Yeah.
I don't know how to pronounce your name. So anyway, thank you so much for giving me such a wonderful introduction. So I'm really happy to share some of the knowledge which I was working in the fermentative biohydrogen production from the macroalgal biomass, and I'm really happy to be a part of it. Thank you so much. So let me start from the beginning. So shall I start? Do you hear me?
Yeah, you can start, please.
Yeah, okay. So as Professor Chow has already explained about my introduction, so I don't need to go further about, so let's start with the sessions. So as this is the introduction about me, so now I'm directly go for the introduction. So why we need to go for hydrogen or any renewable energy sources? As we all know that like we are using the fossil fuels, so human population is increasing a lot, and the consumption of fossil fuels is to be a large quantity, and it's going to be decade soon. So in the recent decades, we are facing a many climatic issues and many environmental problems, and many issues is happen because of like our consumption of fossil fuel is increasing, and it's lead to the excessive damage and totally condemn the environmental systems. So we need to find some alternative energy. So instead of fossil fuels, we need to find some alternative energy that should be much efficient. At the same time, it should be eco-friendly. So in the recent days, we can see that like what is the problem in the environmental system when if it is too much get polluted, or if it is affected by any natural, any global warming or any greenhouse gases effect, so it will damage the entire system, environmental system. As you see, like for example, in recent days, in Korea, we can see that this is a summer actually, but we in the week at least 3 days to 4 days continuously rainy, And in the London, if you see that the temperature is really high, the heat wave is continuously start a lot. So it means like the global warming is the one of the serious issue now that threatening the world. So we need to find some alternative energy that should be very efficient. At the same time, it should be eco-friendly. So that's why the new concept was came to the biotechnologists or the environmental technologists or environmental engineers who are working in the concept of biorefinery. So what is biorefinery? So the nature is giving a lot of abundant source to the humans, like in the way of everything, like for example, the water, the food, the plants, the animals. So the entire ecosystem is always helping the human, which is given by the nature. So from the nature as a biomass, we use that sources to produce various kind of products by using this biorefinery concept. So for example, in the biomass feedstock, if you take like trees, grasses, agricultural crops, and animal residues or animal waste or any waste system, it could be go into the conversion process such as fermentation, anaerobic digestion, gasification, combustion, co-firing. So these kind of process are converting this biomass feedstock into some valuable products. That's valuable products are listed in like the methane, hydrogen, renewable energy, or any chemical, synthetic chemicals, or any. So there are many valuable products is given by from this biomass feedstock when we go for this conversion process. So this entire process is called biorefinery system. So from this biorefinery system, we are going to produce some valuable energy or very valuable energy resources or any valuable energy resources. So as we can see that like in this slide, we can see the feedstock, for example, like the wastewater system or organic waste. So it converted into uh, many products, okay? But particularly, I am going to talk about hydrogen. Okay, so in this case, so how this hydrogen is produced from the feedstock by using various kind of process involved in and produce this hydrogen. So and simultaneously, how this hydrogen is used as an energy resource. For example, like as electricity or fuel, or chemicals. So how this hydrogen is used as an energy source that we can discuss. So why hydrogen? So why hydrogen? So compare when you go for the renewable energy resources. So there are plenty of energy resources are renewable. For example, wind energy is so renewable, and solar energy is also renewable. But as a biotechnologist, as a biological engineer, I'm just focusing on to produce some renewable energy from the biological way. That biological way is particularly from the bacteria. From the bacteria, it produces the hydrogen. So this hydrogen, why hydrogen? So hydrogen, if you see that hydrogen, as a student, you guys may know about hydrogen very well. So it is the purest form of gas as well. And then this hydrogen is having a is one of the best energy carrier. So and then it has when it is combustion to the air, it produces only the water. So this is how, and it has the higher ignition value compared to any other fossil fuels. And this is more than 40 times better than the fossil fuels. So it release abundant of energy and at the same time, but it's really good for the environmental systems. So we can see like, so how this hydrogen is produced in the biological way. So in the biological way, hydrogen is produced by the three steps. One is photolysis, and another one is photofermentation, and another one is dark fermentation. So in the photolysis means by using the algae, for example, like microalgae. So these kind of microalgae will use that light as a source, and then it will produce the hydrogen. And second, we go for photofermentation. In the photofermentation, these organic acids are utilized as a substrate, but however, it also requires some light, and some photosynthetic bacteria will also work on it, and it will break that organic products, and it will produce the hydrogen. But the problem in this both system, in the photolysis and the photofermentation, this process will produce the purest form of hydrogen, but the quantity in the quantity basis, if you think it produces the very low quantity of hydrogen in the both systems. For example, it produces only 0.2 meter cube per meter cube per day. So, I mean, if we think about in the industrial scale level operations, this is a very lowest quantity in photolysis as well as in the photofermentation. And then this in this system also need require a lot of energy resources. For example, it needs some light, and we need to maintain for a long time. And the photosynthetic bacteria are really so sensitive, so it may lead some contamination issues. So there are many kind of issues happening in the photolysis and photofermentation. But when you think about in the dark fermentation, if you take this system, so dark fermentation means the fermentation which occurs without any light. So, and then it converts the organic matters into the valuable added products, such as hydrogen or any other acids or ethanol, whatever. So, as you are a biotechnologist guys, so you may read the TCA cycle, or you may read the citric acid cycle. We can say citric acid cycle, TCA cycle. So in the cycle, we see from the glucose, the final product of pyruvate it produced. So from the pyruvate, there are a lot of ways to go, right? So if you see in this case, same like, so the glucose is such a simple molecule, I mean, simple carbon source that can be break down by the bacteria by producing some enzymes. So that enzymes break the glucose into and produce some valuable other products, and the hydrogen is produced through in this way. So for example, if you see that like in the acetate and butyrate pathway, so the hydrogen will be produced from the acetate and the butyrate. So if you see the theoretical way of calculation, so per 1 mole of glucose, it's give 4 moles of hydrogen, and 1 mole of glucose is give the 2 moles of hydrogen in the butyrate pathway. So why we choose this dark fermentation? So it can be the light sources will be not used in this system, and then there are various kind of advantages. Not only produce the hydrogen, also it can be also produce some various byproducts which will be really applicable in the industry. So that's why we choose the dark fermentation system to produce the hydrogen. So this is the overall flow, which I'm going to explain, like how we produce the hydrogen from the macroalgal biomass, and we use the process optimization technology, and then we use the dynamic membrane concept, and we produce various kind of hydrogen as well as the VFA, and we produce the PHA. That is another topic. So this is an entire system that how I will explain. So why we choose the microalgal biomass? So macroalgal biomass is also one of the abundant biomass which is given by the nature in the sea. So for example, you guys may really fond of Korea culture. So in Korea, we can see the kim, the seaweed. So the seaweed is considered as a macroalgal biomass. So in this case, this macroalgal biomass need the cultivation. You can see in the table the macroalgal biomass harvesting cycle and carbon fixation ability manufacturing process, and there is no disadvantage while we using this macroalgal biomass as a source, and it is really good for this environmental. Not only for this reason I choose this macroalgal biomass, but the another good feature in the macroalgal biomass is this one. So you can see this macroalgal biomass, which particularly the red algal biomass, I am chosen for this study for to produce the hydrogen, and this is abundant source present in South Korea. So usually this macroalgal biomass was classified by 3 types like green, red, and brown. So I choose this red algal biomass. So as you see that, like the one of the main feature I choose this red algal biomass for hydrogen production is because it's contained a lot of carbon source. I mean, for example, if you see in the table, the carbohydrate is present as 67.3% of carbohydrate was present. And among these, the maximum abundance of galactose and glucose was present like 23% to 14%. So this is the one of the major carbon source which is really needed for any microorganisms can utilize this simple basic carbon source of glucose and galactose. So for this main reason, I choose this red algal biomass for my research to produce the hydrogen. So whenever we take any polymer substrates, for example, like this macroalgal biomass or any lignocellulosic biomass. Lignocellulosic biomass is nothing but the nature trees or any straws or any plants which contain the lignin. So any lignocellulosic biomass or algal biomass, it has a complex nature. So when you need to get something simple molecules from the complex nature, you need to break it. You need to break the polymers and you need to change it into the monomers. So to break the polymers, we need to apply various kind of strategy or various kind of things to break these polymers and need to produce or need to get the monomers. So for example, like this pre-treatment, we call this technology as pre-treatment. So for this pre-treatment, there are three types: physical, chemical, and biological. So in the physical method, you can use any radiations or using any autoclave, I mean the temperature, high temperature or waves. This kind of thing can be used to break this complex polymer and into produce the monomer. Or the second thing is like the chemical method. In the chemical method, we use some enzymes or acids or any other alkaline pre-treatment, like these kind of any sources to break this polymer. And in the third way is biological way. In the biological way, we use some fungus or bacteria. That bacteria can also produce some enzymes. That enzymes will break this polymer. But in this case, we use the physical chemical method to break this polymer. Why we don't go for biological method? Because biological method, it's a time process, right? For example, like if we break the polymer into the monomer, this microorganism have to grow in the polymer first, and it need to utilize that sugar, and it need to produce some enzyme. That enzyme that should break that another polymer, and it should be give some monomer. So it's a time process. So for that, to limited of the time, we use this physical and chemical method to break the polymer into the monomer. So, but unfortunately, when we see that like breaking any polymers into the monomers, particularly this carbon, I mean, particularly when it is having a high rich sugar content, when you break it by using any acid method or any heat treatment method, it will surely produce some inhibitors. So that inhibitors, in particularly in the macroalgal biomass, like such as in the macroalgal biomass, which I'm choosing as Geranium amanaceae. So it contains the high, as I said, it has glucose and galactose. The rich amount of galactose was present there. So when we use some acid pretreatment, it produced the major inhibitory compound, which is called 5-HMF, which is 5-hydroxymethyl perfurate. So what this inhibitors will do in the microbial system? So what happened like when these microorganisms, or what is the main purpose these inhibitors are doing in the microbial system, and how it is damaging? So for example, if you see that like the 5-HMF or any perfurate derivatives or any formic acid, limoleic acid, so this will break the DNA of the microorganisms, and it will also disturb the cell membrane. So the osmosis will not be regular. So, and it will completely change the systems in the inner cell organelles of the bacteria. So these kind of things were cause severe damage to the microorganisms. This is how these inhibitory molecules like such as 5-HMF or any perfurate derivatives or this formic acid and limoleic acids are working in the bacterial cell. So now let's go for the parameters. So there are many parameters in hydrogen production. So if you start to produce the hydrogen okay, for example, let me go slowly. Like to produce the hydrogen, there are two kind of main operation modes we need to carry forward. One is batch mode and a continuous mode. So batch mode is for to understanding the microbial system. So I mean, like for example, if you go for any industrial scale level operations or any operations, you started with the batch mode to understand this, how this bacteria system is working and what is the proper environmental have to be given when it is go for continuous operation. And continuous operation is a model for the industrial scale level operations. So this continuous operation is the exact model for the industrial scale level operations, like such as we approach many parameters, how this same the industrial scale level operations also approach in the thing. So we do this one as a lab scale model in the continuous operation. So in this continuous operation, there are many parameters are involved in the hydrogen production, and we can see that HRT, temperature, and pH. So mainly, you guys much more clearly know about the impact of pH and temperature is very important in the microorganism systems. But you may not heard about the HRT. The HRT is also one of the factors called the hydraulic retention time. So, which is very important factor in the continuous operation, where it determines the bacterial microbial growth. So when we go for like in the hydrogen production, so there are two, we need to optimize these HRT conditions. Either it should not be long, either it should not be low. So we need to optimize these HRT conditions by applying various kind of experiments. So if you give a longer HRT in the continuous operation, then this will be not suitable for hydrogen producing bacterial community. It will be suitable for the methanogenic bacterial community. So methane will be produced when you approach in the hydrogen. If the HRT is too short, what happens is, so what is meant by retention? So it stabilizes inside the reactor. So for example, how much, so the hydraulic retention time is nothing but, so when you give the feed, the feed will be go into the reactor and it will be retained in the reactor, and it will be utilized, the substrate will be utilized by the bacteria, and will be the residuals will be came out from the effluent. So the time which it is stabilized in the reactor and utilized for the substrate, which is called the hydraulic retention time. So for example, if the shorter HRT was applied in the reactor, it will be it is not good for the bacteria, and it will release the maximum biomass, and it cause the active biomass wash out. And second is temperature. As temperature, you know, like in many microbial factor, temperature is play an important role for their growth and to produce the optimized condition. So if you see mesophilic temperature is very suitable for hydrogen production, and in some cases they are using some thermophilic hydrogen production by using higher thermophilic bacteria. But however, if you think about in the energy consumption way, mesophilic temperature is obviously around the world, right? I mean, maximum of the countries having the mesophilic temperature. So the mesophilic temperature is one of the best way to produce the hydrogen. And second is the pH. So as you know, like pH is very important factor for all the bacterial growth. And if if the pH is very high, it will affect the hydrogen production pathway. And if the pH is very low, it will be switch the pathway to produce some ethanols and solvents. So these are some of the previous studies by the various kind of researchers study about these kind of parameters, which I say like the temperature, pH, and the HRT, and they showed the maximum of hydrogen production from their studies. So you see that pH around, it should be 5.5 to 6, and then the temperature is always to be a mesophilic temperature, and HRT it depends on various range, but it should not be not longer than the 12. So and then you can see the maximum of the hydrogen production rate, which is termed as HPR. So you can see that that is a hydrogen production rate from these various study between the parameters of pH, temperature, and HRT. So the second thing we discuss about here is about the immobilization. So what is immobilization? So when you may much more aware of it, or you guys may know about this immobilized system. So immobilization technique is used for the bacteria to be stay viable for a long time, or we try to preserve this bacterial cell, and then we need to try to enhance the suitable environment for the bacteria to be present. And so this same immobilization technology we apply our hydrogen system to produce the hydrogen. So there are various kind of materials were used in this immobilization system and to see the hydrogen production. For example, like you see this calcium alginate is the main compound, main polymer, which is trap the bacterial cells, and we use the hydrogen for the entrapment and silicon dioxide for the mechanical strength, and we add the activated carbon to produce some pores. When we add some industrial wastewater applications, it will control the toxicity of the feedstock. So by observing, so we did many research on this to applying the various kind of materials to making this immobilized bead system, and we try to increase our microbial quality to produce more hydrogen production. So this is how hydrogen producing granules will be worked. For example, if you consider this as a whole bacterial granules, and this red color is other dominant bacteria, and this blue color is only the hydrogen producing bacteria. But when we check in the hydrogen producing granules, we try to enhance this hydrogen producing bacterial community in hydrogen producing granules, and we try to stop this other non-beneficial bacteria present inside the HBG. So for this study, we started with the enhancement of biohydrogen production to check the foaming effect. So for example, so once you started any microbial fermentation or microbial production, so if the foaming is the main factor, which can in some cases it will be very useful, but in some cases this is not useful. I mean, the producing foam will affect the biogas rate, and the producing foam will affect the hydrogen production, and in case the producing foam the microbial biomass will attach into the foams, and it lead to the washout of active biomass in the system. So we thought to control the foaming issue in the hydrogen production reactor. So we apply various kind of anti-foaming agents in the batch experiment first, and we found this LS303 is one of the best polysilicon based anti-foaming agent which is commercially available. And so we tried with the various kind of commercial available anti-foaming agents, as well as the natural anti-foaming agent which is called oil. So the natural oils, I mean, when we cooking also, when we see when we cook the meat or any lentils, if the foam is coming, we put some oil in it, then the foam will control. So the same thing, like we tried with the commercial available anti-foaming agents and the natural anti-foaming agent, and we found this LS303 is one of the best anti-foaming agent which control the foaming in the batch experiment. And that same anti-foaming agents we apply in our continuous operation, and it release the maximum of, it does not disturb the entire system. I mean, it does not disturb the hydrogen reactor system, and as well as this anti-foaming agent itself is helping the bacteria to produce more hydrogen in the system. If you see in the figure, you can see the figure 2 shows the increasement of hydrogen production. At the same time, the VFA, the volatile fatty acids produced from bacteria, it's also produced. And then, so from this, we found okay, this is the best anti-foaming agent can be applied in the hydrogen producing system. So after that, we started with the continuous operation in the simple CSTR reactor with any complex structure. As you see, it's very simple CSTR with the connecting with the feed tank, and the effluent will be go through here, and there will be a pH meter and the gas meter, and the power supply unit will be here. So we apply as I said earlier about the immobilization system. So we apply this immobilization system as an inoculum, and you can see the real reactor picture here. So in this case, you see that we tried with the both kind of inoculation system. One is suspended inoculum, and another one is the immobilized inoculum. So as you see that in the immobilized system, the hydrogen production rate is much higher, much better than the suspended reactor system, because as I said earlier about the immobilized system can be provide much good environmental to the bacteria. At the same time, it is really efficient for increasing the cell viability. So the bacteria can sustain in the reactor and produce more hydrogen in the simple CSTS system. But followed by, we can see that like, so this reactor is very easy to handle, and you can see the productivity also much not lower, but however, when you see that if you shorten the HRT to 2 hours or 1 hours, the hydrogen production is getting very loose. I mean, the loss of hydrogen production is because of the active biomass was getting washed out through the effluent system, because there is no any trapping system present in the reactor. So what happens is when we shorten the HRT into very shorter to increase the productivity, automatically the unsustainable bacteria can be get washed out through the effluent. So to avoid this condition, we started to work on USBR reactor, which is called upflow anaerobic sludge blanket reactor. So these kind of reactors mainly apply in the beverage industry wastewater treatment system to give the to treated the very simple organic substances. So if you see that mainly this upflow USBR reactor mainly used in the industrial scale level operations to get more microbial biomass. But at the same time, I mean, even this reactor is also having a various kind of features. I mean, like we can immediately start the reactor with very shorter HRT. At the same time, it will produce the microbial biomass very rapidly. So we can get the active biomass in a very faster time. But unfortunately, this technology cannot be applied in hydrogen production due to when you shatter this HRT, you see a lot of decrease in the hydrogen production, and it will lose the active biomass. So in this case, you can see the 2 hours HRT show the maximum hydrogen production rate of 56 liter hydrogen per day. But when we switch the HRT into 1.5 hours, the hydrogen production is getting lost. So in this case, so for example, in CSTR reactor, it is really easy to handle, but at the same time, it lose the active biomass very faster. In the USB reactor, you see it is really good at retaining the active biomass, and at the same time, it will be very useful for treating the simple organic wastes in the like behavior industry wastewater. But however, when you go for very shorter HRT, it is also losing the active biomass, because we don't have any trapping system to protect these bacterial cells inside the reactor. So that what happens is the next thing is we try to trap the bacterial cells inside the biomass and inside the reactor, and to produce the hydrogen to be more. So we use, you can see in the figure right side, there is a filter. So there is a filter, we used it into the reactor, and we started the reactor with the immobilized system, and you can see some sludge kind of substance was present on the top of the filter. This is the active biomass, microbial biomass, which is settled in the reactor. So for example, in this, you can see in the figure left side, this is a filter looks like, and then when we started with the reactor with the immobilized beads, so all this bacteria will be go and trapped inside. Even when we switch the HRT to the shorter, maximum of the bacteria can be sustained inside. And when we, it can be avoid the washout of the bacterial system in the fixed bed reactor. So if you see in the hydrogen production rate, it produced the maximum hydrogen of 65 liter day of hydrogen compared to this USB reactor when we operated into the 2 hours HRT. That is the same HRT which we operated in the USB reactor, and but in this case it was lost the hydrogen production I mean, the hydrogen production is low, I mean 52 liter, but here if you see in the 2SHRT, it produced 65 liter. So the hydrogen production was increased in this system because it stabilized the active biomass inside. When we check the microbial community analysis, for example, because these kind of experiments all be done with the mixed bacterial community, so we need to see what kind of dominant bacterial species was present inside. And we found, like in the 2SHRT, the maximum abundance of Clostridium butyrium species, one of the known hydrogen producing bacteria species, which produce more hydrogen. So that community bacteria was high, and then followed by other rest of the bacteria. So in this case, we found that, like, okay, this big fixed bed reactor is also one of the very good configuration that can trap the active biomass inside the reactor and produce the hydrogen. But if you guys notice this in this presentation, this R study we done with the very pure substrate of galactose. Why we do with this galactose as a pure substrate in this study, which is really mimicking the macroalgal biomass, which is called the Gelidium amanaceae, as I mentioned earlier. So that has abundant sugar of galactose. So this is kind of a mimicking that substrate of microalgal biomass, and showing like we just apply this pure simple substrate as a galactose as a feedstock, and it produces the maximum of hydrogen 65 liter in the 2SHRT. So followed by when we see, okay, so now we directly apply the pure galactose with, as I said earlier in the pre-treatment, it produced some toxic inhibitors. So that inhibitors, we thinking about, like, okay, so when we pre-treated the macroalgal biomass and we extracted, and we found that that galactose has a inhibitor of 5-HMF, and its concentration is 1.5 gram per liter. So that same concentration we are adding into our feedstock. So for example, from 0.3 gram to 2.4 gram of 5-HMF, we are adding into the reactor feed tank in the period of time, and we see the effect of hydrogen production and the toxicity of the 5-HMF. So if you see this, like until 0.6 gram per liter of the 5-HMF concentration, the hydrogen production was around 26 liter. But once the HMF concentration was increased into 0.9 gram per liter, the hydrogen production was immediately dropped down. So it means like this high HMF is really toxic to the microbial community system. Particularly, this fire is really toxic to the hydrogen producing bacterial community. So we need to try to think about how to reduce the toxicity of the 5-HMF, or to increase the microbial quality to degrade this 5-HMF initially first, and to convert this galactose into the hydrogen production. So after these fewer studies with applying the various kind of reactor configurations, and applying the various kind of strategies, and applying the glucose and galactose as a pure substrate in the study, finally we just apply the direct red algal biomass as a substrate into the hydrogen reactor system. But in this time, we try to store this all the bacteria to be present inside the reactor. So we try some novel inoculation strategy. So what we did is, as I mentioned about the fixed bed reactor, we are trapping this bacteria system into the filter. So the same thing. Now what we do is, so this is like a filter. So we pack all the immobilized bacterial system inside on the pole. So we pack it completely and we kept into the reactor. So what happened is, so most of the bacteria try to stay stuck into the reactor, and it cannot be get to be washed out, or and at the same time it can be stabilize their production performances. So that actually that was our real idea when we did it. But unfortunately, what happened is when we apply this red algal biomass as a feedstock, due to some unexpected error was happened in the feedstock, it totally stopped the feeding and it disturbed the reactor system. So, and this happened again and again. So we cannot successfully completely run this entire experiment to achieve the maximum of hydrogen. But however, until we run this reactor with this particular efficient concept, and we produce the higher rate of hydrogen production of 20 liter per day as a complex substrate compared to any other studies which done by in the previous years. So after this, so after we found like, so this red algal biomass is one of the suitable substrate for the hydrogen production in the fixed bed reactor configuration, which produce 20 liter. And now we again try to apply the various kind of reactor configuration to increase the hydrogen production. And we try to start with the dynamic membrane reactor concept. So what is dynamic membrane? So generally dynamic membrane is nothing but when the membrane system is working on in the reactor, it will be accumulate on the membrane and cause some fouling and increase the sludge quality and making the cake layer of the biofilm. So that is very important factor in hydrogen production, because hydrogen production is happening not only because of only one bacteria or two bacteria. This entire system having a symbiotic relationship. So through this, it will produce the maximum hydrogen. So in this case, we using some membrane kind of technology apply in this dynamic membrane reactor concept, and we call this one as a submerged, because it is we kept inside the reactor. So we kept the membrane system inside the reactor and we produce the hydrogen from this. So we call this one as a submerged dynamic membrane reactor. And as you see in the table, it produced in the 2 hours HRT of 58 liter of hydrogen production. But this is even not equal to the FBR reactor production, because FBR reactor was produced 65 liter. So that is very huge compared to this dynamic membrane concept. But however, this reactor system also one of the efficient reactor system. But due to that, like when we see like some unexpected error was happening in this reactor failure, it caused the failure to the reactor system. So the hydrogen production was getting lower when we switched the HRT into further 1.5. So after we see that there are two, so this is how the membrane reactor system was working. So if you see, this is the real reactor. In the right side, you can see there is a filter model, and we just simply use the polyester mesh as to trap the bacterial biomass. So for example, this is a cylindrical model, and that cylindrical model was covered by the polyester mesh. And we using this is called as a membrane unit. So we particularly say as a membrane unit. So from this membrane unit, so if you guys are, I'm not sure about how you much are aware of reactor operations things, but so we give some recirculation here, and the effluent will be go throughout here. So in this case also, we see the maximum of hydrogen production rate was achieved in 3 hours HRT 57 liter. But when we switch into the 2 hours HRT, the hydrogen production rate was decreased. So, but in all cases, compared to all the reactor configurations, we say the fixed bed reactor configuration is one of the best to produce the maximum of biohydrogen production from the glucose as a substrate. And we check the microbial community analysis for all our samples in all the reactor configurations, and we found like so when the reactor was getting failure in the samples, you can see that like the lactobacillus concentration was a little bit increased, but however, the Clasterium butyricum is the dominant species which is present in all the samples. So from this table, I can conclude to say that like among the various kind of researchers tried the various kind of research on hydrogen production with the reactor configurations. our group, this is what our group was working on with the glucose and galactose as a substrate, and we use the CSTR, FBR, and USBR, and we produce the maximum of hydrogen production compared to the any other previous researchers by using this simple substrate as a concentration, and we produce the 65 liter of hydrogen. Is one of the biggest achievement rather than any other people did as a hydrogen research now, until now. So from this, I can go with the conclusion. Like, so hydrogen, as you know, like hydrogen is uh, one of the best renewable energy carrier in the world, which is really efficient, and at the same time it produces the clean energy. So to produce the hydrogen, we need dark fermentation is one of the best way to produce the hydrogen by using the dark fermentation. And for the dark fermentation, there are various kind of bacterial technology like mixed culture or pure culture need to be used, and the batch experiment and continuous experiment have to be optimized. In the continuous experiment, HRT, pH, and temperatures are one of the factors need to be addressed and need to be optimized immediately. And then the second thing is like, so from this, we tried the various reactor configurations like simple CSTR, USBR, and FBR, and dynamic membrane reactors. And we found that FBR is one of the best reactor configurations, can be produced much hydrogen production rather than any other configurations. And at the same time, 5-HMF is one of the toxic component which is really affecting the hydrogen production. However, it need to be also to be when the future researchers like you people can find some good source or good idea to degrade this 5-HMF in the hydrogen production and improve the system, then it can be acid hydrolysis, that algal biomasses will be one of the suitable substrate for to increase the hydrogen production in the future. Thank you so much, everyone, for your kind attention, and special thank to Professor Chow for your kind. Yeah, thank you.
Thank you, Dr. Bativan, so much about an interesting presentation. Now, I would like to summarize some information about the result.
Yes, sure.
Yeah. This is the result about the optimize the factor in the biohydrogen production.
Yeah.
So the result result about how to control the factor to affect with the biohydrogen production, such as BS, temperature, X, XRT, and after that the research use the bacteria to get to make a reactor to get biohydrogen from the reactor with the reactor configuration, such as CSTR, UASBR, FBR is the factor to affect to the biohydrogen production. The experiment, as you know, the excessive use of fossil-based fuel energy sources cause a severe damage to the environment system, and this is a very crucial moment to find the alternative clean energy sources to control the pollution. So biohydrogen is one of the most promising clean energy sources. Thank you so much for very very interesting presentation.
Thank you so much. Yeah.
And I would like to invite some the question from the lecturer and student from the technology and applied science faculty, please.
Yeah, sure.
Các bạn sinh viên, quý thầy cô có câu hỏi nào cho diễn giả không ạ? Dạ, em xin mời câu hỏi từ quý thầy cô và các bạn sinh viên ạ. Ờ, Chắc trong thời gian chờ đợi thì em cũng xin nói một phần về cái tóm lược về nghiên cứu của cái Dr. Pathiban bằng tiếng Việt. I would like to introduce some information about the research in Vietnamese.
Yeah,
Thank
sure.
you.
Yeah.
Như chúng ta đã biết thì cái việc mà sử dụng cái năng lượng mà gọi là có cái nguồn gốc mà hóa thạch thì đó là một trong những cái nguyên nhân mà nó ảnh hưởng đến môi trường rất là lớn. Và do vậy thì đây là một cái nghiên cứu hiện tại đó thì nó tìm ra được một cái nguồn năng lượng mà sạch hơn và có thể thay thế để mà nó giảm tác động đến ô nhiễm môi trường. Và cái biohydrogen là hydrogen sinh học thì đó là một trong những cái nguồn năng lượng mà sạch và nó có hứa hẹn và nó đem lại những cái lợi ích hơn so với những cái nguồn năng lượng mà hóa thạch mà khác hiện đang sử dụng ở trong cái cuộc sống ngày nay. Thì gọi là cái tảo biển là macroalgae biomass là những cái sinh khối về tảo biển thì được biết nó giống như là các loại tảo mà sống ở trong những cái khu vực là ở biển sâu và những cái khu vực vùng ven biển. Thì những cái tảo này thì nó rất là giàu về cái galactose ở trong cái được thể hiện ở trong cái hemicellulose, và nó được sử dụng trong những cái phương pháp và người ta xử lý cái tảo này thông qua những cái phương pháp gọi là tiền xử lý, ví dụ như là về cái xử lý về axit, và người ta xử lý axit để người ta thu được là cái đường thông qua cái đường hướng chuyển hóa là Ley-lo, là con đường chuyển hóa Ley-lo, tối ưu hóa những cái gọi là những cái đặc tính khác nhau thông qua cái việc sử dụng là cái pH này, HRT có nghĩa là đo về cái tốc độ phát triển của tảo, và cái reactor configuration có nghĩa là những cái tham số trong cái reactor để mà tối ưu hóa được những cái nhân tố ảnh hưởng lên làm tăng cái việc sản xuất ra biohydrogen. Và người ta sử dụng những cái tham số khác, ví dụ như CTR, URSBR, FBR và MBR thì đã được nghiên cứu, và trong mỗi cái tham số như vậy thì người ta sẽ có những cái điều chỉnh để làm sao tối ưu hóa được cái quá trình tạo ra biohydrogen. Và cái đó thì một số cái thông tin cơ bản của bài nghiên cứu thì em cũng muốn nói thêm để cho các bạn sinh viên có thể hiểu được là cái nội dung chính của nghiên cứu. Thì trong cái quá trình mà ông Dr. Pathiban trình bày thì cũng rất là mong quý thầy cô, các bạn sinh viên có những cái câu hỏi để mà mình có thể hiểu thêm về những cái vấn đề này.
Well, you're explaining a lot.
Yeah.
Yeah, nice.
Jeff, please. In the time we are waiting for the question from the student, I have a question. I want to ask you about what is the dynamic membrane bioreactor and effect of it with the biohydrogen production.
Okay, so actually dynamic membrane kind of reactors usually applied in anaerobic digestion. So for example, like in the methane production, to retain the sludge and to increase the sludge activity, they use the dynamic membrane. Dynamic membrane the term is dynamic, means sudden change, right? I mean, suddenly change something in the microbial system. So this membrane, which is applied in anaerobic digestion to retain the active biomass present inside. So that same technology we apply in the hydrogen. So there are no people who was not studied about this dynamic membrane stuffs in the hydrogen. So we try to apply that kind of technology in hydrogen production. But there are still some of the factors need to be, excuse me, so need to be optimized. But however, we achieve the maximum of hydrogen production in the dynamic membrane system that is nothing but there is some cylindrical stainless steel support and it should be covered with some polyester mesh. That is dynamic membrane. So what happened is, so when all the bacteria can be attached into the membrane and it causes some fouling and the maximum it can be retained in the active biomass all inside the membrane unit and the rest of the effluent will become out. So from this factor, we apply the same technology in the hydrogen production, but however, some of the factors cannot be directly really applicable in the dynamic membrane reactor, so it causes the loss of hydrogen production.
Yeah, thank you so much.
Yeah, sure.
Yes. So the dynamic membrane reactor is one of the factor to affect to the biohydrogen production.
Yeah.
Productivity.
Yes, correct. Yes.
This is the most important factor to affect with the biohydrogen production.
Okay, so that's a good question. So hydrogen production is very sensitive, so it can be affected by any factor. So, for example, temperature, the pH, the HRT, the substrate concentration. So this hydrogen production is a little bit sensitive because the bacteria which is involving in the hydrogen production is strict anaerobes.
Yeah, excuse me, in your opinion, what is the most important factor affect to the biohydrogen productivity?
In my thing, I cannot say which is really very, all the factors is very important in hydrogen production. But however, HRT is one of the important factors.
Yeah.
Yeah. HRT is important. Yeah.
Thu nhập từ biển gọi là seaweed. And I want to ask a question. How about the cost? How about the cost for your processing from the collection the seaweed to make the biohydrogen? Is it cheap or is it expensive?
Yeah, that's good. So actually, seaweed biomass is really cheap in Korea. So it doesn't cost much more. For example, within $1, like $1, you can buy a half kilogram of that seaweed.
Yeah.
So from this, you can, I mean, the processed one. So, but I didn't calculate about the pre-treatment cost and the production of hydrogen. But however, compared to the lignocellulosic biomass substrate or any other substrates, red algal biomass was really comparatively cheap than other biomasses which are already available. And it is really abundant source, which is really easy available in Korea. And red algal biomass, it's not a feed for humans. So people not eat that red algal biomass. So it can be used for the research purpose. Yeah.
Is it easy to find the red algal from the Korea? It's easy to
Yes,
find.
yes. So in the south region of Korea, have the abundance of this red algal biomass
Yeah.
in the coastal region. Yes.
No. Yeah. Có thể. Ừ. Dạ, em xin mời các câu hỏi của quý thầy cô, các bạn sinh viên nè. Alo. Em giới thiệu thầy nha thầy. Em giới thiệu thầy nha. Hãy cho bạn biết trưởng khoa.
There are two Vietnam students is also working in our lab now.
Yeah. So this is the experiment we do in the lab or in the field. We do in the lab.
Yeah, actually we are doing it in the lab. So this continuous operation, all we do in the lab. Yes.
Yeah, just collect the agar from the sea coast.
Yes, correct. And then grind it and add the percentage of acid, and we do some heat treatment using the autoclave, and that's it.
Yeah.
And that liquid part we use it as a substrate for producing the hydrogen.
Các bạn ơi, các bạn nông nghiệp công nghệ cao ơi. Alo. Alo. Bạn Bền, bạn Xanh, bạn Khôi, có bạn nào hỏi, có câu hỏi nào không? Các bạn có thể nói tiếng Việt thì sẽ chuyển ngữ sang tiếng Anh để có thể hỏi được thầy Othiban đó. Các bạn. Alo. Dạ. Alo, còn quý thầy cô nào có câu hỏi cho diễn giả không ạ?